last talk is by Pratyush Tiwary. He's I still also maybe associate now, Pratyush, or
no, it's somewhere with the provost or something.
Soon to be associate professor at the University of Maryland, College Park. So he, in the Department of Chemistry, Biochemistry, Technology. So he is for a young scientist that had a distinguished set of awards that for lack of time I will not go through. One thing that I do want to point out is he's a marathon fanatic, and so he's also done a lot of mountain climbing. So let's welcome Pratyush for engaging talk. Thanks.
Okay, is my audio clear?
Yes.
Okay, great. So first of all, huge thanks to Aditya and Bhaskar and some others for organizing this workshop, and especially for the hybrid setting. It's always hard on the organizers to balance everything. And I apologize, I'm not there in person. I had to give some other talks which complicated things, but then also I have had cold for the last few days. So it's not COVID, but you never know, right? So it would have been a mess anyway. So it's the last talk of the whole thing. I will try to not get into too many equations, epsilons and deltas, and try to give you a big picture of what my lab is trying to do in Maryland. I have been here for a little under 5 years, and we have a nice interdisciplinary team made up of students from biophysics, chemistry, applied math, and that's my dog. Since I'm giving my talk from home, he's probably going to show up in the middle of the talk. And we have been funded through a variety of organizations, especially at the moment. We have a career grant and NIH R35 and some DOE funding running, and also the Sloan Foundation grant. And we use computers time from Exceed and wherever else we can get hand on it. So the big picture of today's talk really starts from a book that I'm sure many of us in this audience have read at some point and possibly been inspired to do science due to this book. At least it was the case for me. The Feynman Lectures on Physics, where Feynman says that everything that living things can do can be understood in terms of jigglings and wigglings of atoms. So it's a generic sounding comment, but basically it means if you can simulate how atoms are talking to each other, that's really all there is to life, perhaps. So this is a bold statement, and in the next paragraph, he makes another statement, which is, I think, equally interesting, which is if you knew, for example, the lineup of a protein or of an RNA, you cannot tell what protein is going to be made. He's almost saying that you cannot solve the protein folding problem. So this is Feynman Lectures, which is 1963. We have come a long way from there. Already in 1964 and even earlier, people were coming up with these molecular dynamics and Monte Carlo methods. So the first molecular dynamics papers were Alden Wainwright, 1959, and 1964 by Anisur Rahman, also from India, where, on a big supercomputer, they were able to simulate how 864 atoms talk to each other and move. And Metropolis, Monte Carlo is even older. So it's been 60 years since all of this. Where are we now? So, well, it looks like we are from those 864 atoms, we are now doing almost billion atom molecular dynamics. This is the spike protein of coronavirus from Romy Amaro's lab. It was a full 2-page article in New York Times. And AlphaFold took all of us almost by surprise last year, if you really think about it. And it made to cover of Nature magazine, and not just Nature, it was even in popular press everywhere. One morning I woke up and the news article in my Google News is Forbes magazine saying AlphaFold is the most important achievement in AI ever. That's a really bold statement, and I mean, it's not very common that folded protein structures make it to Forbes. So this really begs the question, are we there yet? Have we really solved all of this through to achieving Feynman's vision? So the answer is we have made a lot of progress, and this is my view, but we still have a long way to go. First of all, in molecular dynamics, while we are simulating bigger and bigger systems, we still have a timescale problem where you simulate the system, most of the time it is trapped in one state. And the reason is that when you are solving molecular dynamics, you have a timescale, it's a differential equation with femtosecond time step. So with supercomputers, such as the one in Maryland's campus, you can get to maybe 100 nanosecond. With GPUs, you can start pushing them even more. With really specialized computers, such as Anton, made by D.E. Shaw, you can get to millisecond. But to study a lot of problems that my lab is interested in, and many others are, such as crystal nucleation or protein ligand dissociation, the timescales are prohibitively slower. So there are these enhanced sampling methods where you can speed up the simulation, you can accelerate it. The idea being that if you know what is the so-called reaction coordinate of the system, which is really a smart, low-dimensional reduced representation, then you can start to fill up bias in the system. You are essentially doing importance sampling. You're filling up, you're keeping memory of where you went, and next time when you go there, you're less likely to get trapped there. And through this, you can do back and forth. So these methods exist. A big problem in these methods is how do you figure out what is a good reduced representation or a good reaction coordinate without having solved the problem to begin with. And there is a cottage industry of these enhanced sampling methods with acronyms that I really don't expect in this audience, or even in my computational chemistry audience. Most people are clueless as to so many methods keep coming up all the time. So that's molecular dynamics. Secondly, AlphaFold. It's made a lot of noise and it's very useful, but there are huge problems. First of all, it does not have a notion of Boltzmann diversity. When you look at the protein, it's not just about the ground state, it's also about excited states. What are the other conformations a protein can take? And often the ratio of this ground and excited state changes with simple mutations in the protein. You make a single mutation, the ratio changes completely. You make a second mutation, the ratio completely flips. And what are these excited states and what is the ratio of ground state and an excited state has huge repercussions for drug design and problems like that, and for just simply understanding how our body functions. And for these three sequences, for example, I mean, with AlphaFold, it's become so easy to use it. All my biochemist colleagues these days think they are like hardcore programmers, living inside the matrix. They just go to collapfold, put in a sequence, get a structure, and you can put these three. It's really, really easy. And turns out AlphaFold completely gets it wrong. For all three of these, even though the mutation is completely disturbing the landscape, it has no clue how it's being disturbed. So you might say, well, it was not in the training database. If you put it in the training database, then AlphaFold would get it right. But then the point is, if in order to get something, you have to put it in the training, is it really a prediction? Where is the extrapolation? Or is it just smart interpolation? Which is a question really perhaps for all of AI. And so my lab aims to develop methods to solve these problems and some other grand challenges. Today, I really won't have the time to go into all of this. I will probably mostly talk about just this information bottleneck method. I will tell you a little bit about what we are doing with LSTMs, and that's it. And you can check out all our codes on GitHub. So the reason why we are inspired to develop these methods is, first of all, it's a lot of fun to really play with AI at the interface of physics and chemistry. But also with this, we can solve challenges which I think are relevant to not just scientists, but to the whole society on its own. First, for example, even in proteins, AlphaFold, while it has made a lot of progress, it has limitations. But proteins are not just all there is to biology. We also have RNA. And turns out that while the number of known human RNA is much more than 10 times the number of proteins, the number of structures in the protein data bank that involve RNA is 1% of the number of protein structures. And the number of drugs FDA approved that can actually target human RNA is a grand total of 2. That's it. So after AlphaFold came out, there was another competing method using geometric deep learning, which got a much lower resolution than AlphaFold for RNA. And they had to train only on 18 structures. That's it, because even out of these 1,500 structures, most of them are RNA complicated with protein. That's it. So pure RNA structures are even lower. And yet they've made it to cover of science. So if we really want to come up with an alpha RNA, my prediction is we are at least 50 years away. This is so hard of a problem. Similar problem applies to alpha phase transitions and so I was originally trained as a metallurgist in India at one of the IITs, and I'm very interested in how, dislocations nucleate or thin films grow, which is perhaps a common interest with Bhaskar, and how crystals nucleate. And this is somewhere where material science and biology join together, because if you want to predict which neurodegenerative diseases can happen, all of them, one of the common theme is basically some form of a phase transition, almost crystallization, where the proteins start to aggregate in one direction and that's it, and it becomes irreversible. If this process keeps going on, you can get Parkinson's, Alzheimer's, all these diseases, and you can never really come back from it. So these diseases tend to be incurable, and there is a reason for it, because this phase transition, when it happens, it's irreversible. So all these challenges are the things that I would like to solve over the next coming decades along with the community. And for this, we have to start by developing very, very basic methods. And there are a lot of problems in developing such good methods. We have to think about better featurizations. In molecular sciences, the data sets that we have are very different from data sets in social sciences. We have structure in the data, so people are starting to use graph neural networks to think about it in smart manners. I'm not going to talk about this today. Mostly I'm going to talk about this sampling problem and how my lab is integrating theoretical progress with method development and experiments and AI at the same time. So let's start with the sampling problem. The method that I'm going to use, or my lab developed, is actually really simple. It's just a form of active learning where you have molecular dynamics. This in case is a RNA, it's a riboswitch, and you have a small drug-like molecule dissociating from the RNA. So if you do molecular dynamics, not much happens because of the timescale problem. So you pass it through something that looks like an autoencoder, and we will come back to this. It's more like a time-lagged autoencoder, but let's get back to this in a moment. But something that can reduce the dimensionality and learn the predictive features of what's going on here. So you can use this reduced dimension and do these sampling methods that I mentioned early on, where you're biasing the landscape along these reduced representations. And now you are getting superior sampling because you have done important sampling along some degree of freedom. At this point, don't publish, take this superior sampling, learn a new reduced representation, and keep iterating. So these ideas have existed in different forms. Some of you are probably familiar with, many of you are probably familiar with diffusion maps, where it is the same idea. You learn this low eigenvectors of a diffusion map, you sample along that, and you keep iterating. So the challenge though is there are so many reduced representations, and not all reduced representations carry physics. Some of them do, not all of them do. Let me show you a simple example. This is a 6-state system where you have states numbered. Look at the 2D picture, the one on the bottom. So you have 6 states, 1, 2, 3, 4, 5, 6. And you can come up with a reduced representation where you cluster everything just along R1. You remove all information along R2. So now forget about the 2-dimensional picture. Just look at the 1-dimensional picture. It's beautiful because it's giving you all 6 states. You have not lost any information about the states. However, it's quite wrong because it's telling you that from state 4, you can go to 1 and to 2 with equal probability, which is simply not true, right? You can, from 4, you can go to 1, but in order to go from 4 to 2, you have to go through 1 or to 5. So information is lost. So this is just a simple projection. Over the years, a lot of reduced dimensional methods have come up. Of course, PCA is the granddaddy of these methods. There are so many others like TSNE, SketchMap. And in my community, at least, people sometimes tend to put too much trust in reduced representation. We get nice clustering and we think that's it. It can be used to drive the sampling. So this is where we have to be careful. And that's where I want to introduce to you this information bottleneck concept. This really goes back to the idea of rate distortion theory from Shannon. And it's a classic paper from like early 50s that I highly recommend reading, but it got more popularized in this label of information bottleneck by Bill Bialek and others. And then my group has been using it in chemistry, and we are discovering how this information bottleneck has actually deep connections with theoretical chemistry. So what's an information bottleneck? The idea is really simple. Let's say you have some high dimensional input and some output. The output could be also high dimensional, or the output could be just a set of labels, right? So for example, the input could be the temperature of all cities in the United States today, and the output could be the temperature of all cities tomorrow. Or the output could be something as simple as, is it going to rain in the city of Ames tomorrow? Just a binary thing, yes or no. Or in our case, the input is a molecular dynamics trajectory of a system until a certain time. And the output is, what is this trajectory going to look like in the future? So information bottleneck says that a good model should have 2 properties. First of all, it should be as predictive as possible. If you're not doing a good job of predicting, what good is your model? At the same time, it should be as compressive as possible. Compression is more abstract, but I think hopefully this audience will appreciate why we want the model to be compressive, because if the model is not compressive, then it's really learning everything. It's committing everything to memory. It's not figuring out the signal from the noise, right? So you get this bottleneck structure where you want to compress as much as possible while still being able to predict as well as possible. You can write down this trade-off between compression and prediction as the difference of 2 mutual informations, and you introduce a Lagrange multiplier here. Or a control parameter or a regularization parameter. If beta is equal to infinity, then you simply don't care about prediction. You're just trying to compress as much as possible. If beta is equal to 0, you don't care about compression. You're just trying to predict as much as possible. Interestingly, if you set beta is equal to 1 and the input the same as the output, then you recover a variational autoencoder. So this information bottleneck is a generalization of a variational autoencoder. And there is a bit of math into how we really optimize it, but the idea is now quite simple. We have a molecular dynamics trajectory. We want to learn the future of this molecular dynamics trajectory. We express both the encoder and the decoder functions as deep neural networks. And in this paper in Journal of Chemical Physics, we showed that, so in chemistry, which is my real field, people have very well-defined notions of what is the best reaction coordinate, what is the best mechanistically meaningful reduced representation. It is known as a reaction coordinate, which even if you don't do chemistry now, you have probably seen the name reaction coordinate in your high school textbooks in chemistry, where you have a two-state, you have energy wells, and you always have the x-axis marked as reaction coordinate. So in this paper, we show how this has real connections with the reaction coordinate in chemistry. And there's a bit more nuance here when we implement it. This bottleneck variable, most systems in chemistry are not a single state system. There is a mixture of states. So why use a simple Gaussian prior? Here we use something called VAM prior, also from Maxwelling. So instead of having a single Gaussian, we approximate that as a mixture of Gaussians. And the number of Gaussians that we need is adjusted on the fly. As we keep iterating through this method, we find that many of the initial states, they disappear into one state, and we recover how many states does this system have. So this simple idea of, let me go back 2 slides actually, of taking a trajectory, learning a past future information bottleneck using VAMP prior. Here I am showing you a linear encoder. You can start with you can also use a nonlinear encoder. We thought nonlinear encoders should be more powerful. Turns out for most problems, when your initial data is not very reliable, it's best to stick with a linear encoder, because it regularizes the model. If your data is not trustworthy and you too quickly jump into a nonlinear encoder, you can end up overfitting noise. So, which is something which was counterintuitive to us. So with this framework, we can keep iterating now just between molecular dynamics and machine learning, and we can do a lot of fun things. For example, we can fold and unfold small proteins. On the left, you are looking at a peptide, which can be left-handed or right-handed, and it's in all atom water, so I'm not showing you the thousands of water molecules. And we can, it's a slower than millisecond process, and we can do it by learning this reduced representation on the fly. This is connected to what something the last speaker mentioned. It's similar to approximating the Koopman operator here. So this information bottleneck is also an approximation to the Koopman operator, strictly speaking. So we are learning that from very limited or very biased data and improving it on the fly. This in another small protein. Here you're looking at how a small ligand permeates through a membrane. We can look at real anti-cancer drugs and explain as to why certain mutants lead to resistance from the drug. And the answer here is that if you make a certain mutation and the drug spends less time in the protein, and I'm really jumping gears here and going into biochemistry, but the big picture here is that if you can calculate how much time does a drug spend inside a protein of interest, you can do a very good job of predicting how effective is the drug going to be in really the patient surviving. So with these type of methods, we can really get the predicted residence time of the drug in very good agreement with experiment, not just for one system or 2 systems, but really a ton of systems. Well, 7 is not ton, but we have more systems here, and other people are also using the methods. And this is really across 12 orders of magnitude. So these are molecular dynamic simulations where the experimentally expected residence time goes from nanoseconds to hours. And our methods in all atom molecular dynamic simulations, by learning the reaction coordinate on the fly and speeding it up, can give these predictions. And we do better than experiments, right? It's not just about getting one data point in agreement with experiments. We do better than experiments because, first of all, there are huge error bars in experiments. As computation and theorist people, we tend to put sometimes too much trust in the experiments, while experiments have their own sometimes a priori unknown error bars. So we do better than that. We have quantified error bars. And more importantly, experiments can quantify how much is the residence time. They cannot tell us why is the residence time what it is. These all atom simulations are really showing the mechanism of why is the residence time what it is and what should we do to make the drug stay there longer. And we can also look at RNA. This is different ligands in an RNA. And here we can predict which drug, which mutation. So even if this is too much biochemistry for you, none of us, I mean, no one in the audience, I'm ready to bet, has not heard the word mutation in the course of the last 1 year, right? It's in fashion these days, sadly, due to COVID and things like that. So for this RNA molecule, we can predict for drug number 1, which mutation will have more impact, which mutation will have less impact. And for drug number 2, similar things. So in this case, we predicted for drug number 1, this is actually not a drug, it's just a small ligand and it's a small RNA molecule, it's a model system. We predicted that this mutation will affect this drug more than the one, the mutation on the bottom, and vice versa for the other drug. And then our collaborators at National Cancer Institute, which is not too far from here, went and actually predicted our mutations. And those of you who know, like quantum mechanics would appreciate this. This is quite a lot of distance, right? So in quantum mechanics, we call it spooky action at a distance, right? It's so far. Why should a mutation over here affect a drug that goes and binds over here? So just on the basis of structure, our collaborators could never have predicted that this mutation would have a role. It's really have to simulate the thing and see how it dissociates in order to make such predictions. And again, switching gears, we can not just biology, we can also look at problems such as crystal nucleation. This is urea molecules nucleating in water. This is a process which takes not even minutes or days or hours. This is days and weeks long time scale process. It takes a lot of time to happen. And you can see these beautiful crystals forming, but then the thing is, it's not just one crystal, it can form these different polymorphs. And for urea, you might not care what the polymorphs are, but for pharmaceutical drugs, it matters a lot. For example, paracetamol, it can exist in 2 different crystal forms, and you can see both of them forming over here. One of them will help your headache. The other one, at least it won't cause you headache, but it won't help your headache. It's useless. This is glycine. It's the simplest amino acid that exists. Really, really small molecule. Turns out in one polymorph, it is an antidepressant, and in other polymorph, it doesn't do anything. So these are all atom simulations in water molecules where we can simulate these things and really talk about processes, which if you were trying to do on a supercomputer, it would take you possibly the age of the universe to get a single event. And so that's all about molecular dynamics. Before I switch gears again, any questions about this part? About the rave method?
Pratyush, quick question here. How is the autoencoder? How do you relate that the retina representation with the reaction coordinates? I'm curious. Is it a constraint that you place, or is it something that comes organically out?
If by organically you mean half a page of math, then that's yes, correct. So you have to see this paper, and I'm really skipping that. But especially with the use of VAMP prior, we can show that it approximates what is known as the committer, which is the true reaction coordinate. So with the use of VAMP prior, we can show that a past future information bottleneck, which is not a true autoencoder, it is more like a time-lagged autoencoder, is indeed the true reaction coordinate. So I can give you a bit more intuition here. So an autoencoder takes the input and tries to reproduce the same input, right? So here, this is a state autoencoder. You take the input and you try to predict what is the state of the system. And let's add one more layer of complexity. You don't try to predict the state of the system at a certain time t. You try to predict the state of the system at certain time into the future. And that's why it's a reaction coordinate. So you have a bunch of atoms in a protein. You take all of it, an autoencoder would just reconstruct the protein. That's not a reaction coordinate. What we are saying here, take all the atoms in the protein and tell into the future, like few picoseconds into the future, whether the protein is folded or unfolded. If your time-lagged state predictive autoencoder can do this, then it becomes a reaction coordinate. Hopefully that answers the question.
Thank you. Yeah.
Okay, so let me go to the second part and then we can come back to questions. So this one connects to really looking at time series coming from experiments, and we have seen this in this workshop in many contexts. So our inspiration was just like we do autofill, which really mesmerizes me, especially this part where it said, let's get together soon for tacos, right? It's highly non-trivial because the last time taco was mentioned was so many alphabets and so many words ago in the subject line. So if you think about this as a generalized Langevin equation, it's really keeping memory from way back in the past. And that's how our brains also are, right? What I'm going to tell you next might be influenced by a conversation I had yesterday, or perhaps 5 years ago. It can come and influence what I'm going to do next. So our idea was that could we use these autofill type trajectories to really look at dynamics coming from biomolecular systems or even general molecular systems theory or experiments, and use autofill on this type of trajectories to show what's going to happen next. So, and yeah, sometimes I tried to type this on my phone and it came up with some funny things. It's kind of amusing. So we use long short-term memory network for it. I always get asked the question, why not transformers? There are two reasons. Number one, the machine learning field moves so quickly that it's hard to keep up with methods. And number two, my group, we try to stay away from methods where we cannot really look at them from a statistical mechanics perspective. So long short-term memory networks were simple enough that in this paper, we could show that training an LSTM is actually equivalent to learning the path entropy of the dynamical system. So, and this works quite nicely. This is an experimental time series on pulling a riboswitch, switch, where if you look at the extension data coming, raw data, it looks something like this. It's highly non-Markovian. What this means is that if you're over here where my cursor is pointing, you don't really know what's going to happen next, because you might be coming from this basin on the top, or you might be coming from this basin on the bottom. So it's highly, highly non-Markovian. But with LSTM, we can do a good job of reproducing such trajectories. And we really outperform competing models such as hidden Markov models for this. And yeah, the time series are identical. You simply cannot, so we can train an LSTM on a given time series and then start generating copies of that in a very quick and cheap manner, which you simply cannot say whether it's true or wrong. So this is all good, but it's perhaps too good. And the reason why it's too good is because AI suffers from garbage in, garbage out. So this is the synthetic peptide that I showed you earlier. So bear with me as I explain this to you. So this peptide is known to be, so it can be a left-handed helix or a right-handed helix, And turns out that if you really did a very long simulation on this system, you would find that both populations are symmetric. It's not a chiral molecule. And sorry about that. So, but if you did a short MD simulation, you would find that there is slight thermal noise. You get a slight differentiation in the probability distribution. So now if you train an LSTM on this, it's like an undergrad who tries to copy from the homework and does not quite realize what is good and what is bad, and copies every little thing. So you can pick up, oh, this is wrong, you copied, right? So this is what happens over here. If you train an LSTM on this, it copies it identically. It preserves the same noise. Now, instead, if you had run 10 times longer molecular dynamics, you would have seen that it becomes symmetric. So how do we solve this problem? You might say, well, just add a constraint, right? Add the constraint that things are symmetric. And which we saw in many talks. It's definitely useful to inductive biases and things like that. LSTMs are a bit tricky because what you're doing here is train a recurrent neural network. It's a dynamical system with very, very long feedback. So when you try to add constraint to an LSTM, it's not as simple as just adding a Lagrange multiplier and getting the job done. It's complicated. So for this, we were able to come up with a method called path sampling long short-term memory, and where the idea was to really exploit the finding we had in the first paper about this in Nature Communications, where we showed that the LSTM is learning the path probability. If it's really learning the path probability, then we start adding constraints in the space of paths. And I'm going quickly here, but I want to show you a result of how it works, and you can read the preprint on this to get a better taste. But essentially, this method now allows us to really control the probabilities in the space of paths, also learn the right Lagrange multiplier for doing it. And now, for example, for this system, if you had, and then again, I apologize for really skipping. This is really, really new work. We submitted the preprint like only this week. So you have a short MD simulation or an experiment where if you just train LSTM, it preserves the asymmetry. But with this method, you can really tune the LSTM and start generating dynamics which conforms to whatever constraint you introduced into the system. This is for an MD simulation. You can do it for really fancy things. This is a quantum dynamics simulation where you are observing the number of photons coming out of a cavity. And same thing here. This is dissipative dynamics. So you're seeing how many photons are coming out. And you can go and what you can control here is the photon rate, the rate of emission for the dissipative dynamics. So it's very interesting dynamics because early on there are these Rabi oscillations and then it dissipates. So you saw the dynamics for a given dissipation rate. You can train LSTM on this. It will do a wonderful job. But now you can use path sampling LSTM to generate a trajectory for another dissipation rate for which you never did the fancy quantum experiment. And if you did do that experiment, you would find that this PS LSTM would have generated a trajectory which looks just like that. So this is all. So I think I'm going to wrap up now. So I showed you mostly about this RAVE method where we learn an information bottleneck, which I claimed is the reaction coordinate. With this, we can speed up the molecular dynamics, do back and forth. I showed you a little bit about LSTMs, especially the path sampling LSTM, where we can learn non-Markovian dynamics and start to infuse physics into the problem, which is very useful because a lot of time series are undersampled. We know AI is wonderful if you have lots of data, but for many practical problems for society and engineering, we don't have lots of data, we are limited, and where it's important to infuse data. I really did not talk about these denoising diffusion probabilistic models. You can read this on arXiv, which is also a lot of fun. And all the codes are on GitHub. And with this, I think my group, we are really excited about solving problems, just programmable thermodynamics and kinetics, and really, doing self-assembly where we are not just talking about what phases will form, but when will certain phases form. This is not my cover, this is just some cover from soft matter. And we are getting in a big way into the RNA folding problem and really try to predict which human sequences are targetable by human RNAs. And then with my collaborators, experimental collaborators at NCI, we are starting to predict which ligands can then go and bind with this. With this, thank you very much for your attention, and I will be happy for any questions.
Any more conversations?
So in the autoencoder framework, do you fix the time lag?
Oh, great question.
How much does the sampling?
Yeah, sure. May I know the name of the person asking the question?
I'm thinking about the previous speaker.
Okay, great. So this is a wonderful question. So we have this past future information bottleneck, and we don't know how far into the future should we predict, right? So there are these methods known as Markov state models, where the idea is predict so far into the future that the dynamics becomes Markovian, so you lose all memory. Here, how far into the future we predict is really a control parameter. So the lag time is a fast mode filter. And the simple idea is that if you predict too far into the future, you wash out most of the states, right? So for this little biomolecule called alanine dipeptide, if you're predicting 2 picoseconds into the future, if you remember what I mentioned about the VAMP prior, we start with 10 states, and if you predict 2 picoseconds into the future, 8 of them disappear, and we are left with only 2 states. If we predict half a picosecond, we are left with 3 states. So we don't really have to select how fast into the future we predict. It's really a filter which allows us to show how coarse-grained we want the system to be. So hopefully that answers your question.
Thank you. Yeah. For the reaction coordinate approximation of the autoencoder, in that case, is the time lag?
Yeah, that's a good point. So there, we really don't know which time lag to pick. So what we do is to, there we don't have a very good answer to this problem because we don't want to just speed up 2 states, right? We want to speed up most of the states, right? So we really, sorry, there are some kids outside my house trying to distract me. So there we start with some, we have to make a choice and put it through RAVE and see whether the next round it improved or not. So there it's an open question. So if you're really trying to understand a system, then the time lag, you can make different choices. But in the context of RAVE, where you're trying to speed up the simulation, we start to go with a longer lag. And if it starts speeding it up, we stick with it. If the system gets trapped and iterations do not speed up the simulation, then we start reducing the time lag, and that's what we do. But there are some open questions here, which hopefully mathematician friends can help me answer better.
All right, let's thank the speaker for